Vô lên cuộc họp á, thấy được 3 đứa, mình tự thấy
Mẹ
Tắt tiếng chị ba.
Xin được chào mừng toàn thể các quý vị đại biểu đã tham gia hội thảo trực tuyến tăng cường khả năng phát hiện biến chủng Omicron bằng xét nghiệm sinh học phân tử ngày hôm nay. Hội thảo của chúng tôi sẽ bắt đầu sau 5 phút nữa.
Chị Hà về rồi ạ?
Chị về rồi ạ.
The deadly coronavirus. Coronavirus. Economic
The lockdown has been expanded.
In early March 2020, the World Health Organization declared COVID-19 a pandemic. As more and more people became ill and economies began to shut down, clinicians and scientists started working together to find answers. It became vitally important to develop a diagnostic test, which is precisely what the scientists at Thermo Fisher Scientific did in record time. Our real-time PCR test, which diagnoses the virus in less than 3 hours, was authorized by the United States FDA for emergency use and later received the CE mark in Europe. The Applied Biosystems taqpath COVID-19 test kit is now available in nearly 180 countries. Why was this kit so rapidly adopted for use around the globe? Because regulatory bodies throughout the world know what we all know, that to combat the pandemic, we need accurate, proven, gold standard technology. We need to work on a global scale. We need scientists to accelerate innovation with urgency. And that's why Thermo Fisher Scientific is leading the world in COVID-19 testing. Our scale and depth of capabilities have never been more vital. And our mission to enable our customers to make the world healthier, cleaner, and safer has never been more important than it is today. From the beginning, Thermo Fisher mobilized supply chains, activated R&D resources, and deployed field teams to enable our customers to meet the demand for COVID-19 testing all over the globe. From New York to Los Angeles, Sydney to New Delhi, Rome to Frankfurt. We know how important it is for you to have access to reliable supplies for COVID-19 testing. And it's our top priority to support you during these challenging times. Even as parts of the world continue to shut down and face other COVID-19 challenges, we deploy local support teams to keep your labs running, anytime, anywhere. When on-site visits are not ideal, our service and support teams are there to guide labs remotely. With our industry-leading supply chain and world-class service and support, Thermo Fisher is leading the world in COVID-19 testing. 50% of COVID-19 testing is performed on Thermo Fisher technology, and we have the capacity to manufacture tens of millions of tests per week, making us your trusted supplier for your ongoing COVID-19 testing needs. Get your reliable supply of COVID-19 test kits today at thermofisher.com/COVID-19.
Xin được chào mừng toàn thể các quý vị đại biểu đã tham gia hội thảo trực tuyến tăng cường khả năng phát hiện biến chủng Omicron bằng xét nghiệm sinh học phân tử của chúng tôi ngày hôm nay. Công ty cổ phần thiết bị SISC Việt Nam xin gửi lời chào và lời cảm ơn chân thành tới sự quan tâm và hợp tác của quý vị trong thời gian qua. Công ty cổ phần thiết bị SISC Việt Nam là đại diện phân phối chính thức của hãng Arm Torrance Applied Biosystem Thermo Fisher Scientific Mỹ tại Việt Nam. Kính thưa quý vị đại biểu, đại dịch COVID-19 ngày càng diễn biến phức tạp. Virus SARS-CoV-2 liên tục xuất hiện các biến chủng mới, gây khó khăn trong công tác phòng chống dịch. Ngày 26 tháng 11 năm 2021, WHO đã xác định biến chủng B11529 hay biến thể Omicron là biến chủng đáng lo ngại. Đây là biến thể có số lượng lớn các đột biến, trong đó có nhiều đột biến trên gen S có thể liên quan đến khả năng lây truyền và khả năng trốn thoát hệ miễn dịch cao hơn. Với mong muốn trao đổi, cập nhật thông tin các giải pháp tăng cường khả năng phát hiện biến chủng để sẵn sàng ứng phó với dịch bệnh COVID-19, công ty chúng tôi kết hợp với hãng sản xuất Thermo Fisher Scientific tổ chức hội thảo trực tuyến tăng cường khả năng phát hiện biến chủng Omicron bằng các xét nghiệm sinh học phân tử. Nội dung chương trình hội thảo hôm nay bao gồm phần trình bày của đại diện hãng Thermo Fisher là ông Christopher về các giải pháp sinh học phân tử của hãng Thermo Fisher giúp tăng cường hỗ trợ phát hiện và nghiên cứu biến chủng Omicron, bao gồm có phần trình bày về giải pháp realtime PCR, giải pháp điện di mao quản hay giải trình tự Sanger và cuối cùng là giải pháp giải trình tự gen thế hệ mới NGS. Theo sau là phần thảo luận được dẫn dắt bởi tiến sĩ Phạm Thu Hằng, Giám đốc Trung tâm CGG Center. Bài thuyết trình đầu tiên chúng tôi muốn giới thiệu với quý vị trong buổi hội thảo ngày hôm nay có tựa đề Biến thể Omicron, các thách thức trong việc phát hiện và sàng lọc biến thể SARS-CoV-2 bằng xét nghiệm real-time PCR do ông Christopher trình bày. Để thuận tiện cho việc truyền tải thông tin đến quý vị trong thời lượng ngắn gọn của buổi hội thảo hôm nay, chúng tôi xin phép được trình chiếu các bản thu mà ông Chris đã dành riêng cho buổi hội thảo ngày hôm nay, kèm theo phần phụ đề tiếng Việt.
Good day, ladies and gentlemen. This is Christopher Ong, a regional technical sales from Thermo Fisher Scientific. Just a quick introduction of myself. Prior to joining this company, I served as a medical technologist at Molecular Diagnostics Center, National University Hospital of Singapore. I have more than 7 years of experience on validating and running clinical molecular diagnostic tests, covering infectious diseases, mutational analysis, heritable diseases, as well as leukemia. Given that clinical molecular diagnostics have gained prominence recently, especially during the COVID-19 pandemic, the emergence of new SARS-CoV-2 variants poses new challenges not only for COVID-19 detection, but also variant screening and surveillance of public health. I'm happy to be here today to share with you some key updates about the Omicron variant and how do we address these concerns through molecular diagnostics using our real-time PCR solutions. Before going into the content, let me first briefly introduce you the company I am representing, Thermo Fisher Scientific. Our mission is, with the wide spectrum of life sciences solutions, we aim to enable our customers to make the world healthier, cleaner, and safer. In terms of our teams, we have established global thought leadership in all areas of science and harnessing the power of all that knowledge, with more than 10,000 customer-facing reps, so we can always be there for our customers. No matter what science, we have focused experts and scientists in-house that are focused on the same things. We are also committed to advancing science and staying ahead of innovation through research and development, generating more than 25 billion of revenue per year. As you can see, our work has been largely business to business, serving the society through our partners. Today, with the COVID-19 pandemic, we have started doing so in a more direct way. Thermo Fisher is at the forefront of the global response to COVID-19, leading the enablement of our partners in the fight against the virus. We support the research of the virus, diagnosis of infection, providing essential lab equipment and personal protection equipment, as well as supporting development and production of new treatments and vaccines. Today, over 50% of COVID-19 testing are performed using the Thermo Fisher technology. When the coronavirus outbreak began in late 2019, Thermo Fisher Scientific rapidly developed one of the first COVID-19 diagnostic tests, which has now scaled to manufacture 20 million tests per week. Thermo Fisher Scientific is working to apply its skill as the world leader in serving science to standardize and industrialize the manufacture of tests and testing platforms in a reliable and reproducible way to support the global need for testing at scale. Globally, over half of all COVID-19 testing is currently being performed on Thermo Fisher Scientific instruments. We have rapidly increased our production of COVID-19 kits and manufacturing capacity to 20 million reactions per week and beyond if needed. Labs have been constrained with supplies from reagents, consumables, and instruments. Thermo Fisher will provide assured supply for your lab to ensure that there are no concerns in your workflow and throughput. For COVID-19 testing, Thermo Fisher Scientific is vertically integrated. Reagents, instruments, the uncontrolled spread of COVID-19 globally has contributed to conditions that favors virus evolution. Multiple reports have also shown that the virus has been mutating at a rate of about 1 to 2 mutations per month, especially for some recently detected variants mutated even faster beyond scientists' expectation. What might new SARS-CoV-2 mutations and variants mean? And why should we worry or be concerned about that? These variants might increase the speed of human-to-human transmission, change the severity of the disease, affect the susceptibility to therapeutic agents, evade vaccine-induced immunity, as well as impact detection by clinical tests. Therefore, we have this classification of SARS-CoV-2 variants that helps inform public health, lab, and enhance community response. In terms of variants of interest, they are known to carry genetic changes that affect virus characteristics, such as transmissibility, disease severity, immune escape, therapeutic, or diagnostic escape, with community transmission with apparent epidemiological impact to suggest an emerging risk to global public health. While variants of concern would meet the criteria of variants of interest, these variants have been demonstrated to intensify the transmissibility or deadly change in COVID-19 epidemiology or decrease in effectiveness of public health. For example, the Lambda and Mu variants have been assigned as variants of interest, while before that, we have the Alpha, Beta, Gamma, Delta as variants of concern, including Omicron being the latest addition to this category. Let us look at some of the responses from public health organizations and government. On November 26, global public health institutes like WHO, Africa CDC, European CDC, as well as US CDC all released official classification and statements about the Omicron variant. Following that, some countries quickly enforced travel restriction or border closures, expediting national vaccination programs. Confirmed Omicron cases were already being reported in more than 20 countries back then. This figure highlights our response to SARS-CoV-2 Omicron variant. If you look at the timeline for Omicron emergence and spread, the variant was first detected in South Africa in late November, followed by a call with CDC 1 week later to discuss about S-gene target failure or S-gene dropout observed from our PCR test. We then launched 2 Omicron-related mutation genotyping assay in the first week of December. There were more than 26,000 cumulative Omicron sequences detected in 81 countries as of December 2021. As we observe further on the Omicron sublineage trend and the 6970 deletion on the S gene of SARS-CoV-2, the BA1 strain still dominates the Omicron landscape, with 99.5% of Omicron sequences detected. This figure in red also show that the number of samples with S6970 deletion increases steadily over time within just a month. As mentioned, Omicron has spread to 81 countries so far, with most countries becoming the predominant variant. This figure adapted from GIS8.org shows the distribution of Omicron sublineages detected by country, dominated by the dark blue dots or the BA1 sublineage. There is also a rapid surge in the number of Omicron sequences observed in countries like the UK, Canada, and Australia within just a few weeks of time. With that, I would like to introduce you the taqpath COVID-19 assay, which we designed carefully with mutations in mind. By mapping to more than 55,000 SARS-CoV-2 genomes, we included only the most stable parts of the genome and avoided unstable regions. We identified and targeted unique regions of the SARS-CoV-2 genome to avoid detecting other coronaviruses with 100% homology to all SARS-CoV-2 isolates, primer, probe sequences of operating frame 1AB, N gene, and the S genes to ensure optimum specificity. We built in redundancies with multiple gene targets to compensate for virus mutation at the primer sites, which has been useful in detecting and understanding both the alpha and beta variants. Of course, now for the Omicron variant as well. This figure simply clarifies how we chose targets in the most stable areas of SARS-CoV-2 genome, with the lower risk of mutation at the time of design. Again, these 3 target design functions to compensate for virus mutations. Let us now discuss further about S-gene advantage and what it means in Omicron variant detection. As mentioned, the B11529 variant, or commonly known as the Omicron variant now, first reported in Botswana and South Africa in November 2021. So far, what we know about this new variant is that it carries a large number of concerning mutations with more than 30 genomic changes. This includes the 6970 deletion on the S gene, as well as 15 mutations in receptor binding domain. The Omicron virus may transmit faster, reduce vaccine effectiveness, and increase risk for reinfection because of these genomic changes. In this case, the WHO published a general guideline that contains recommendations on how we could deal with this new variant through enhanced surveillance, actively report cases, including field investigations and lab assessments. This press release also highlights an important comment from the director of the Center for Epidemic Response and Innovation in South Africa, that Thermo Fisher tests allowed them to detect cases that may contain the Omicron variant through S gene dropout. This early identification will be critical in helping to track and understand the spread of the Omicron variant to South Africa and the world. Attached is the WHO technical brief that indicated Thermo Fisher taqpath assay can be used as proxy for the Omicron variant before sequencing. The S gene target failure approach may lead to faster detection rates. The European CDC also recommended the use of Thermo Fisher assay as a screening method for Omicron. This is yet another urgent briefing that reiterated the utility of S gene target failure feature as a screening tool for the Omicron variant. This was strongly supported by an early evidence that 100% with S gene target failure were the Omicron variant. Furthermore, European CDC also reported that the S gene dropout can signal the presence of 6970 deletion, potentially the Omicron variant, especially in settings that are still dominated with the Delta variant. This approach will quickly help to differentiate patients on whether they are infected with the Delta or the Omicron variant. I would like to just briefly highlight the summary of variants and our test here. We have released and been updating this reference guide to notable SARS-CoV-2 variants based on WHO classification to inform our customers about the latest known impact on our PCR test. As you can see here, that we have included all previously reported variant of interest or concern. In this case, we have indicated the S gene advantage, which means the S gene dropout may signal the presence of 6970 deletion and potentially the Alpha variant, and now the Omicron variant. We will now spend the next few minutes to discuss about research tools available for SARS-CoV-2 mutation and variant surveillance. Epidemiological surveillance is conducted to ensure viral diseases match the reference strain, as well as to monitor possible mutations. This is important as many changes in the viral genome can impact public health policies and options, how the illness spreads in the population, potential study of treatment options, and vaccine development research. There are still many unknowns about SARS-CoV-2, which need to be understood to better predict virus strain evolution. Scientists will need to constantly ask questions like, how can we detect new variants? What is the origin of a new variant and how prevalent it is? Will current molecular or antigen-based tests miss detecting a variant? Or does the variant spread more quickly or lead to higher disease severity? And last but not least, what is vaccine efficacy against a new variant? That is why we aim to become your trusted partner in SARS-CoV-2 surveillance. For strain surveillance, we offer multiple research solutions through next-generation sequencing or NGS, or Sanger sequencing to help sequence the full viral genome or targeted regions of interest. When scientists look to confirm and verify known mutations associated with specific strains or lineages, They could use either our Sanger sequencing or RT-PCR genotyping solutions. In terms of detection, we have been offering a reliable, accurate, and high-quality COVID-19 testing through RT-PCR, helping you to assess emerging mutations' impact on performance claims. Again, we want to be your single trusted source for service and support by ensuring you a secured supply chain that meet your testing demand. Lastly, I would like to also introduce to you the Taqpath SARS-CoV-2 mutation panel, which offers convenient, customizable real-time PCR research solution for detection, analysis, and surveillance of the SARS-CoV-2 variant. You may choose from a menu of verified real-time PCR assays to build your own custom SARS-CoV-2 mutation panel and get the results you need today while preparing you for what is to come. These scalable solutions lets you to run a few or hundreds of samples to identify one or many mutations, all on your current real-time PCR instrumentation. The workflow for our mutation panel is fairly straightforward. As you can see, following your regular PCR detection workflow, you may move to the confirmation protocol by using the same RNA extract to confirm mutations associated with the SARS-CoV-2 emerging variants. We have quickly launched a few made-to-order assays also to help streamline single nucleotide polymorphism genotyping of the Omicron as well as the Delta Plus variant. I hope this session gave you an overview for today's webinar. We can't wait to share more details with you in the next 2 sessions. You may visit thermofisher.com/COVID-19 mutations to learn more. With that, I thank you for your attention.
Xin cảm ơn ông Christopher về phần trình bày trên. Nếu quý vị có câu hỏi nào cho phần trình bày vừa rồi, xin mời nhắn câu hỏi vào mục chat box. Chúng tôi xin phép được giải đáp trong phần thảo luận. Bài báo cáo tiếp theo có nội dung về giải pháp điện di mao quản hay giải trình tự Sanger để giám sát, định chủng và phát hiện biến thể Omicron.
Hello, my name is Steve Jackson, and I'll be your host for the 4th and final module in the SARS-CoV-2 surveillance protocol descriptions. In this final module, we will be discussing the protocol that we designed that will allow customers to sequence any position in the SARS-CoV-2 genome by Sanger sequencing. We'll present the browsers that we've used for finding primers, and also a brief description about the data analysis tools that we've presented for customers as well. So what we've done in these 4 modules is provide 4 different protocols for SARS-CoV-2 surveillance. In the first 3 protocols, or in in the first 3 modules, and I think it was in the 3rd one, we discussed the 3 protocols that we previously released for sequencing the S gene dropouts, for focusing on B117 and B1351 strains, and for sequencing the entire S gene and the S gene alone. This 4th protocol that I described for you is the most flexible, and it's geared towards letting customers sequence any place they need to in the SARS-CoV-2 genome. We've described for them tools that allow them to pick up the primers they need for sequencing, for running the chemistry, and for if they have variant reporter or SeqScape for doing some variant analysis and identifying if any of the mutations that they identify in their experiment map to the mutations that have been identified in variants of concern. And so we hope this gives customers the most flexibility to identify strains present, either existing strains or new ones that might arise, and help control the progress of the pandemic. So thank you very much. This protocol is meant to complement the other ones that we have posted on our website at the beginning of the year. In particular, this protocol gives customers the flexibility to choose primer sequences that will sequence any region of the virus, not just those found in common variants or the S gene. This is because new variants will arise all the time, and customers need to be flexible in order to sequence the regions where these new mutations arise. And so this protocol provides two tools for choosing wet lab-tested Sanger sequencing primers that can be used to sequence any place, any location, any coordinate in the SARS-CoV-2 genome. Included in these tools are primer sets that are listing the variants of concern. These are preloaded into the tools, and customers can then quickly download the primers that are associated with the designated variants of concern or variants of consequence. And if we check the list of variants of concern in the tools every 2 weeks and update them as needed. The protocol also provides details for sequencing with big dye direct and big dye exterminator chemistries, as the other protocols did. And we also provide in the protocol starting information and basic files that can be used for variant analysis using Variant Reporter or SISC software. The protocols and tools can be found here. The website for the Sanger sequencing page is at the top, and that Sanger sequencing SARS-CoV-2 research page then has links to all the other protocols in addition to this one. Within that page is the links to the visual browser and the lookup table. Both of those then can be accessed from that Sanger sequencing page. So the basis of this protocol, or the basis of this tool, is based on two web-based primer lookup tools that we designed. One of them is a visual browser, and the other one is basically a lookup table. We'll have more to say about that in a second. In addition, though, this protocol also includes a detailed protocol that's written to use the lookup tools to find primers necessary, and also use the big dye direct and big dye exterminator sequencing kits. We have some quick reference guides that allow our customers to perform variant analysis using the results they get from the protocol. And finally, on our web page, the Sanger Sequencing Solutions for SARS-CoV-2 Research, we will have links for the protocol, for the browsers, and for the variant analysis protocols, and all the associated files necessary for that as well. Everything should be on this one page. So to start off with, we'll talk about the visual browser. This viewer will allow the customer to look at the entire SARS-CoV-2 genome. The viewer was built using the JBrowse backbone and should be able to work in any browser. The visual browser is basically broken up into three different boxes highlighted with yellow. On the leftmost side is a list of the available tracks that could be loaded into the viewer. On the top long rectangle is a navigation tool that allows you to scroll back and forth across the genome, zoom in and out. It shows you which reference sequence has been highlighted. It allows you to enter coordinates based on the NC045512.2 genome standard, and also allows you to use a highlighting tool if you need to highlight particular regions of the genome. Finally, on the bottom large square is the actual feature viewing area. By the way, this particular browser will be preloaded with the Wuhan original strain, the NC045512.2 reference genome. So to see features in the browser, you basically choose which tracks you want to visualize. In this first example here, we have the genes and coordinates box checked, and therefore in the viewing area on the browser, you can see that we have the different genes that are in the SARS-CoV-2 genome annotated. You can see they're kind of like those bars with the names of the genes below them. In the bottom browser now, we have clicked on the B117 variant track. In this case, this track has been preloaded with all of the different mutations that have been defined as part of the B117 strain. The browser will become preloaded with variants that are of concern to the CDC and outbreak.org. outbreak.info. And therefore, the customers can visualize the locations of mutations in variants of concern. by clicking on the different pools, so CDC pool 1 through 6, as shown here on this slide, the positions of the different Sanger sequencing primers can be seen. Here we have a region between about 13,000 and about 16,000 highlighted, and here you can see the positions of the different primers that have been designed to Sanger sequence these particular regions. You can see there's both a left and a right primer that can be used to Sanger sequence across that particular region. Most of these primers are built to be in pairs that can sequence between 500 and 600 nucleotides about. Although in some cases you will see you can maybe customers can maybe choose non-numeric pairs to sequence smaller regions if they want to. In any event, choosing the pool tracks will place the primer positions in the viewer sequence. By the way, the pools that are present here in this example are not the ones going to be in the final viewer. That is upcoming. So the actual tracks and the pools will look a little bit different from this, but basically the way it works will be the same. I also want to emphasize that all the primers that appear in this browser will have been wet lab tested, and only the primers that have worked will be loaded into the browser for our customers. We have found that some of the primers that were designated by the CDC did not perform very well in the Sanger sequencing protocol that we designed. So we designed some new ones, and those will be the ones that are loaded into the browser when appropriate. On the bottom now, we can see that the view, if we zoom in quite a bit, I think this is the most zoomed in view here. You can see the close-up of the sequence with the colored bars, the colored nucleotides. You can see the amino acid sequences in all 6 reading frames. The browser in the final form will have only one of the either forward or reverse reading frame shown, so the customers can see the translation. In addition, you can see that the 2 primers, the SC2M139 left and SC2M138 right, are also visible here. And as you can tell, these are from 2 different pairs, the 39 and 38. The arrows will point to the direction of the primers, 5' to 3' orientation, and so you can use the 39 and 38. 39 left and 38 right primers to Sanger sequence, but it'll produce only about 120 nucleotides of sequence there, and might not cover the region to pick the Sanger sequencing primers that are a numeric matched pair. So to find primers that will cover a particular mutation, what we've done is you can highlight multiple tracks here. So what we've done is we've highlighted the B117 track and the CDC pool primer tracks along with the gene coordinates. The genes are indicated on the top of the file here with the bars. You can see the C14676T synonymous mutation in the B117 mutation, or in B117 strain in this view. And you can also see that the 2 primers, SC2M138 left and SC2M138 right, will sequence across the region that covers the C14676T mutation. And so by choosing those 2 primer pairs, the customer will be able to then design or use these primers to sequence and detect the mutation in the B117 strain. By doing this across all the different mutations or the different coordinates the customers want to use, then they can find the primers that are of interest to them. To get the actual primer sequence then, what they have to do is they have to click on the feature. Any of the features that are here can be clicked, and a window like you see here will be shown with the name, the position, the length of the sequence that's there, and the in the box down at the bottom you'll have the FASTA file showing the sequence itself. Here, the SC2M138 left sequence is highlighted in the light blue. It's only 25 nucleotides long, so it's no problem getting that into one window. And then to order this primer, then the customer would have to copy that primer sequence from the FASTA file, or download the FASTA file, and blast it or put it into an oligo ordering program or something like that. I should also note that this visual browser, although it has the primer sequences, they are not M13 tagged, and if customers want to order M13 tagged primers, they'll have to paste the M13 sequences in manually. The other browser that we have is a text-based lookup app. Like the visual browser, this lookup app can be used with any browser, and it allows our customers then to enter the information in the boxes shown here in order to find the primers that are appropriate for their experiment. So one of the ways that or a couple of the ways that the lookup browser can work is by entering particular strain names or gene names, amino acid changes, genomic coordinates, primer pairs, and they can add the M13 tags to their choices, as you'll see in a second. What I have highlighted here are the strains, the B1351, 117, etc. These strains, the mutations in these strains are preloaded into the browser, and if the customer wants to order all the primers necessary to sequence B117, they can do that very quickly here. The other option is by gene, and so, for example, if customers want to order all the primers that would allow them to sequence the N gene, they can choose that the N, make the gene N choice in this screen here. I also want to note here that this is you can only see one of these two drop-down menus at a time. The customers will not be able to visualize both menus as shown in this slide. This is just for example illustrative purposes only. As I mentioned, this lookup table will allow customers to type in the genomic coordinates. Here the customer has typed in 24567 as the location in the reference genome that they want to query. And then when they hit the search tag and clicking the M13 box, then they will then get a list of the primers that can cover that particular coordinate. Here there are two sets of primers, SC2M162 left and right, and SC2M163 left and right, that will allow the customer to sequence this particular region. Either of these should be able to work. When customers want to look up particular amino acid changes, like for example the E44K, which is commonly seen in some of these highly transmissible variants, they will need to put in the gene name and the amino acid change. For example, here we know the E484K is in the S gene, and so they choose the S gene 484K. And then when they click on search, then the primers that will cover that particular information will be found. Again, here we have the SC2M158 left and right, which allow the customers then to sequence across the coordinates that encompass the E484K mutation. So when customers are ready to export their primer choices, they can click on the Export to Order button. And what will happen is that the browser will then generate a text file loadable in Excel, which will have all the primer sequences, the names, the synthesis scale, and the purification choices already made. All the customer will have to do then is to type in their name, in this case Jane Doe, and then the text file is then uploadable into our oligo ordering page. We have links to that on the Sanger sequencing webpage, as I alluded to before. Note here the synthesis scale is preloaded by default at 25 nanomolar, and the purification is desalted. If the customer wants to change that, they can, although they would have to check to make sure that the scales and the purification choices are correct for our oligo ordering service. Nevertheless, the 25 nanomolar scale and desalted is entirely appropriate for sequencing reactions in this protocol. And the 25 nanomolar scale will produce a large amount of primers and can be used quite a bit for several hundreds to maybe 1,000 different sequencing reactions. Okay, in addition to the protocol and the browsers, the fourth module includes a guide to variant analysis using two of our software packages. One of these is Variant Reporter, and so we have written a brief user guide that allows customers then to take their sequencing results and see whether or not any of the variants that have been defined in the variants of concern are present. So we provide a brief user guide. It's a brief user guide for customers to follow. We have a text file that contains the mutations that are commonly seen in variants of concern that can be uploaded into Variant Reporter. We have a text file that is of the primer sequences that are in the protocol. And finally, there's a FASTA file for the virus reference sequences. All of these will be downloadable on our web page, and customers can then load these into Variant Reporter according to the directions that we have in the protocol. I want to also point out that the user guide that we supply is just a getting started guide. It is not meant to be comprehensive or a complete description of all the features available in our software. Customers who want to have a complete description of all the features should look at the original documentation that came with the software when they purchased it. One of the other important things that is often done with variants is that the sequences that are made are tracked by various global entities like GSAID or the Genome Detective. And so we also provide in the guidelines instructions for exporting the results from these experiments so they can be uploaded then into external databases for tracking and surveillance globally. Again, we don't have direct links into these different sites, but we do provide a guide for customers to export the data that they generate so that they can be uploaded to these external sites for tracking purposes. So I just described Variant Reporter. We have very similar sets of things for SISC for customers that have SISC software. We have the quick reference guide, as I described, for them that allows them to get started. We have supporting files that will be provided that allow, it's a demo project that has the reference sequence, primers, and the variants of concern mutations preloaded into this demo file so that customers basically just have to download the demo file, and they can then upload or interpret their own sequencing results in the SISC environment. And like I mentioned, we also have some guidelines for exporting data so it can be uploaded into external databases. The last thing I want to mention is throughput. We often get asked about what the throughput for these different protocols are with the PCR, the Big Die Direct, the Big Die Exterminator, and CE. And so I've put together some guidelines here for throughput on our different platforms, on SISC Studio, on 3500, on 3500 XL, and on the 3730 XL. With the exception of SISC Studio, all these have a 50 centimeter capillary installed. So to amplify the sequencing amplicons, it takes about 1 hour to run that, no matter what the instrument is. And then for the cycle sequencing and Big Die Exterminator, it takes another 2 hours. So a total of about 3 hours to do the PCR and cycle sequencing and cleanup. And then running on the different CE instruments takes various amounts of time. In general, for running a single set of reactions, a single injection takes about 40 minutes on any of our platforms. And so, for example, on a SISC Studio, I see that per sample it takes about 3 hours and a minute to run, and that's not quite right. It should be 3 hours and 41 minutes to run a single sample on SISC Studio. On SISC Studio, to run an entire 96-well plate, it takes about 20 hours or so. On a 3500, for single sample, it takes about 3 hours and 44 minutes, et cetera. And so you can go through this table and find out, how long it takes to run either a single sample or a single injection, or a 96-well plate that's completely loaded up with samples. And then based on that, we calculated, how many samples a user might be able to run in a shift, on an 8-hour shift, that is. And so if you keep the SISC Studio going, the customer a customer can run about 14 samples in a 96 well plate in an 8-hour shift. That includes the amplicon PCR, the big dye exterminator cycle sequencing, and the CE running time. On a 3500 with an 8 capillary, it can run about 27 samples in an hour, or in a shift. On a 3500 XL, about 81 samples in a shift. And on a 3730 XL, that is 96 well, 96 capillary array installed there, the customer can run about 320 samples in an 8-hour shift if they keep the machine fed. In other words, they would have to set up multiple 96 well plates over the course of that 8-hour shift in order to achieve this throughput. So, and these are just guidelines. Some of the actual performance might vary a little bit.
Xin cảm ơn sự quan tâm và các câu hỏi quý vị đang gửi về ban tổ chức. Chuyên gia từ Thermo Scientists sẽ cùng thảo luận với chúng ta ngay sau bài thuyết trình cuối cùng này. Ở mục tiếp theo, chúng tôi xin giới thiệu về giải pháp NGS trong nghiên cứu và phát hiện các biến chủng SARS-CoV mới, trình bày bởi cô Tan Boy Tran, giám đốc sản phẩm của Thermo Fisher.
To the presentations of fast confirmations of Omicron variants with the ion implicit SARS-CoV-2 Insight Research Essay brought to you by Thermo Fisher Scientific. Before we begin, a quick introduction about myself. My name is GC. I'm the product manager representing Thermo Fisher Scientific. Prior to joining Thermo Fisher Scientific, I have more than 7 years of experience in the provisions of scientific applications, in-depth technical support, and customer training for real-time PCR, capillary electrophoresis, microarray, and next-generation sequencing covering the Asia-Pacific region. First of all, I would like to provide a fast facts on the Omicron variants. The Omicron variants, or B11529, is a SARS-CoV-2 variant. It was first identified in the South Africa. It has been designated as variants of concern by World Health Organizations. Omicron has an unusually large number of mutations and appears to be highly transmissible based on some early reports. The emergence of the Omicron variants has already led to a wave of lockdowns and travel restrictions to mitigate the alarming spread of the variants. One of our Thermo Fisher Scientific customer, who is Fisher here, her name is Dr. Alison Glass. Dr. Alison works in Lanchurch Laboratories in South Africa. She is running the implicit SARS-CoV-2 solutions with the Genexus integrated sequencer, and she is one of the first to discover the fact that there might be a new variant circulating in the community. Dr. Allison was collecting PCR samples from mid-November, and she observed S-gene dropout in PCR results using our applied biosystem SARS-CoV-2 PCR kits, suggesting a possible variant. By sequencing these samples collected from November 12 to 20 in-house with the Ion Torrent Genexus integrated sequencer, Dr. Allison and her team were able to analyze and identify the specific virus mutations in less than 24 hours and alert the South African National Surveillance Program for confirmations and immediate actions. The B11529 reported to WHO on November 24, and on November 26, B11529 is designated as variants of concerns and named Omicrons. In summary, it took about a week from the detections of B11529 sequence to the declarations of an Omicron variant, which is a record time much faster than we have seen for any of the reports of other variants. Part of the reason is due to the fact that she has the GeneXus instrument available in her lab to enable her to rapidly sequence those samples and get results within 24 hours. The variants of concern Omicrons belongs to the Pango lineage B11529, characterized by 45 to 52 amino acid changes, including 26 to 32 in the spike proteins compared to the reference strains. The Omicron has a long list of mutations, some 50 in all. Of these, 32 pertain to the virus spike proteins, a vital part that the virus uses to gain entry into the host cells and which is also the target of most COVID-19 vaccines. Part of the reason why it was concerning is because the number of mutations in the spike protein. Omicron has 32 mutations versus 13 mutations in Delta. There's a lot of scrutiny around the spike protein because it is essential for the virus ability to infect cells, so lots of disease-fighting antibodies can neutralize the virus targeting those spike proteins. If there's a mutation that changes the amino acid in a way that can alter the structure or chemistry of the proteins, it can prevent the antibody from binding as they normally would. However, no one knows how multiple mutations work together. Researchers are still studying it. The implicit SARS-CoV-2 Insight Research Assay is an enhanced assay to survey the complete SARS-CoV-2 genome for epidemiological investigations. The assay consists a total number of 242 unique amplicons across two pools, with 237 amplicons specific to SARS-CoV-2, with the highlight of the additions of variance tolerance primer for emerging variants, as well as 5 newly designed human expression control targets for sample confirmations and quality assessment. The assay covers over 99% of the SARS-CoV-2 genomes. The assay amplicons range from 125 to 275 base pair with significant overlap. It is this overlap that gives exceptional performance over time against naturally occurring variations. In the case of the Omicron variants, I'm happy to share that our ion implicit SARS-CoV-2 Insight Research Assay can enable the confident identifications of the Omicron variants due to the robust coverage provided by the variance tolerance primers. Here's an example to showcase the capability of the implicit SARS-CoV-2 Insight Research Assay in obtaining the complete sequences for emerging variants with the intelligent panel design. In this figure, the upper parts refer to the coverage of the Twist Biosciences SARS-CoV-2 control 1 amplified with our original SARS-CoV-2 variants, SARS-CoV-2 or community panels. And the bottom part refers to the coverage of the Twist Bioscience SARS-CoV-2 control 1 amplified with our latest implicit Insight Research Assay panel. With these twist control viral variants that has a mutation in the original panel primer region, you can see that we managed to rescue the library amplicons with the Insight panel due to the additions of the variance tolerance primers, resulting in the improved coverage. In this study, we are looking into the performance of the implicit SARS-CoV-2 assay relative to competitor assays. Result shows that ion torrent has a higher percentage identified bases in submitted sequence, indicating higher tolerance for new mutations due to the intelligent panel design, which reduce the number of amplicons dropouts. When a base cannot be called based on the sequencing data, instead of A, T, C, or G, an N is inserted. We can quantify how well an assay is performing by comparing the percent of the identified or non-N bases. For US submitted ion torrent genomes in GSET, we see an average of 99.85% of bases identified, compared to 98.83% in competitor assays. As demonstrated in the box plot on the right under the figure 6, we also see much fewer low quality or high-end samples submitted using the ion torrent technology compared to our competitor, which have a much wider range of the percentage, none and. Despite the manifold increase in the number of available genomes, the panel continues to successfully amplify and assemble whole genomes. This study here is based on the data collected from the Omicron sequences submitted in GSET back in 2nd December 2021. The raw data are available in SRA, which is another public database of the SARS-CoV-2 sequences, where key mutations were not identified in 61 out of 279 Omicron samples because of the dropouts caused by the Arctic version 4 protocols and primer set. As you can see from the table here, although ion torrent technology submitted less sequences back in December compared to competitors' technology, we have a very low percentage N for no-call, which is 0.35% as compared to competitors which reported 1.12% and 2.25% respectively of the average N percentage. With a high degree of functional redundancy, the panel has continued to prove that it is robust to the natural variations, allowing for overall sequencing quality higher than competitor assays. In addition, the performance of the assay was confirmed by multiple sites as the intelligent design of the assay provides coverage to identify Omicrons as well as previous VOCs and VOIs. In extent to the previous slides, in this graph, we have tabulated how many times each sequencing technology was able to call either wild type, mutant, or no call for the spike mutants. Again, ion torrent technology provides best coverage of spike proteins with average 1.85% of no call permutations in the spike proteins as compared to 8.58% and 6.61% respectively in competitors' platform. This information is particularly important as the Omicron has a long list of mutations, some 50 in all. Of these, 32 pertains to the virus spike proteins. Here we showcase how ion torrent technology leads the sequencing performance with the lowest percentage of no-call in spike proteins. With our implicit SARS-CoV-2 Insight Research Assay, we provide the comprehensive informatic suite for seamless informatics and data uploading to the public SARS-CoV-2 data repositories. After the sequencing run is completed, the analysis through this plugin are automatically initiated, providing a complete workflow from designs to interpretations, analysis, and data upload with minimal hands-on time. The plugin includes the SARS-CoV-2 coverage analysis and variant caller, which provides statistics and interactive visualizations of coverage over targeted regions of the SARS-CoV-2 reference genome, as well as SNP and indel calling at a given positions. For various annotations, we have the SARS-CoV-2 annotate SNP-F. These plugins text the variants detected by the variant caller and use the SNP-F to produce a list of annotated variants and predict the effects of the genetic variants on genes and proteins with reference to the public or the private database. We also include the plugin to generate consensus sequence. The generate consensus plugin align reads to the reference, identify low frequency variants, and eventually provided consensus sequence in FASTA format. And you can then opt to upload the FASTA sequence to public repositories for global surveillance. In addition, we have integrated the lineage assignment plugin. This plugin assigns a SARS-CoV-2 pangol lineage to its consensus FASTA sequences output by the generous consensus plugin and the pangolin program. Monthly updates on this plugin is expected as well to keep the database up to date. Here's another customer success story featuring Dr. Eva. She sequenced the first Omicron case in Czech Republic. We have a great quote from her talking about the use of the implicit SARS-CoV-2 insight research assay together with the ion torrent genexus integrated sequences, which enable her to confirm the new Omicron strain in less than 24 hours. We do have a blog post here, provides more insights into customer success story on the use of implicit SARS-CoV-2 insight assay to sequence and identify the SARS-CoV-2 variants. For example, the work of Dr. Ellison, which we mentioned earlier, who is one of the first to detect these new variants at Lancet Laboratories in South Africa, and Dr. Eva's work in the detect of the first Omicron case in the Czech Republic. There are more ion implicit SARS-CoV-2 resources available in our Thermo Fisher website. You can go to the browser and type in thermofisher.com/SARS-CoV-2 insight. In this webpage, you can access to more information and NGS educational resources for SARS-CoV-2 research, the ordering informations, recent customer talks and webinar, as well as our customer experience and success story. If you do have any questions, please reach out to us and we are happy to hear from you. And we have also included the links to the additional resources in this slide, including the documents released by the WHO and ECDC on the classifications of the Omicrons, the implications of emergence and spread of the Omicron for the EU or EEA, as well as the Africa Centers for Disease Control and Prevention statement regarding the Omicrons. There are also links to the press release, blog posts, and user guides in regards to the topics of applied biosystem taqpath COVID-19 test and the ion implicit SARS-CoV-2 insight research essay. With that, I thank you for your attention for listening to these presentations. Have a nice day.
Vâng, thưa quý vị, đó là tất cả các nội dung diễn thuyết trong buổi hôm nay của chúng tôi. Nếu quý vị quan tâm và muốn nhận tài liệu liên quan đến buổi thuyết trình ngày hôm nay, xin mời đăng ký nhận thông tin qua đường link trong chat box. Tiếp theo chương trình hội thảo, tôi xin trân trọng kính mời tiến sĩ Phạm Thu Hằng, Giám đốc Trung tâm CGG Center sẽ dẫn dắt phần thảo luận cùng với đại diện của hãng Thermo Fisher Scientific là ông Christopher Ong. The next part of the webinar, I would like to introduce Dr. Phạm Thu Hằng, the director of CGG Center, will be leading the discussion along with the representative of Thermo Fisher Scientific, Mr. Christopher Ong. Xin kính mời tiến sĩ Phạm Thu Hằng và ông Chris.
Hi. Xin chào các anh chị, các quý vị. Như các anh chị vừa thấy là các anh chị vừa được nghe 3 bài trình bày của hãng Thermo Fisher. Thì tôi thấy ở trên này cũng có khá nhiều câu hỏi. Tôi xin tóm tắt một chút về các cái bài giới thiệu ở trong này. Thì từ phía Thermo có giới thiệu 3 bài, một bài về liên quan đến kỹ thuật về realtime PCR, bài thứ 2 liên quan đến giải trình tự sang G và bài thứ 3 liên quan đến NGS. 3 kỹ thuật này là 3 kỹ thuật phổ biến và kỹ thuật chính trong sinh học phân tử để sử dụng để nghiên cứu cũng như là phát hiện SARS-CoV-2. Tuy nhiên cái buổi trình bày của chúng ta hôm nay thì tập trung chủ yếu liên quan đến việc phát hiện biến chủng Omicron là một biến chủng mới và cũng được sự quan tâm của rất nhiều nơi. Vì vậy mà các bạn như các bạn đã thấy trong bài trình bày đầu tiên về kỹ thuật realtime PCR thì Thermo có giới thiệu đến bộ kit realtime PCR là taqpath. ở đây thì họ sử dụng 3 gen mục tiêu là gen N, gen S và ORF1. Như họ đã trình bày thì họ có nói rằng là nếu như mà gen S mà không phát hiện được, không có tín hiệu thì có thể liên quan đến cái việc là đấy là một cái tín hiệu để nghĩ đến đó là biến chủng Omicron. Và để xác định tiếp theo thì chúng ta sẽ phải làm giải trình tự. Thì tôi xin tóm tắt cái ý chính mà tôi vừa được nghe như vậy. Nếu các anh chị và các bạn có câu hỏi gì thì xin mời các anh chị có thể tương tác trực tiếp với cả Christopher ở đây, hoặc là các anh chị có thể viết trên chat và tôi sẽ hỗ trợ để dịch cùng với Christopher. Hi Chris.
Hello, hi.
Can you hear me? I see there's quite lots of questions on the chat box.
Yep.
So I will translate into English to you and please answer the question, please.
Yeah, sure.
So the first question from Dương Ngọc Thúy. Right, so she asked, when we use real-time RT-PCR and with the target gene is E gene, so if the result, if it's Omicron variant, it can have the result is negative or low, positive or not. So Omicron is affect the result for E gene or not.
Okay, so first of all, thank you for the question. I hope all of you benefited from today's webinar. Okay, so to answer your question, in most instances, the E gene will still be detected on the samples containing the Omicron variant. So while the S gene is frequently not detected for the Omicron sample. So for example, if you have a PCR test that covers the E gene or the N gene, for example, you will likely have both results detected. But if you have your PCR test with the S gene as the target, you may not detect the S gene in that PCR.
Okay,
Yep.
thank you for the answer. So tôi có thể tóm tắt cái câu trả lời của Dr. Chris ở đây là nếu như mà đối với những biến chủng Omicron thì gen E vẫn có thể được xác định được trong cái phản ứng real-time RT-PCR. Nhưng mà đối với gen S thì thông thường là sẽ không phát hiện được đối với biến chủng Omicron. So the second question from Hồ Thị Hồng Nhung, biến thể Omicron tác động như thế nào đến độ nhạy của xét nghiệm PCR? Okay, the second question is for the Omicron variant, is it affect the sensitivity of real-time PCR or not? Please.
Okay, so there will be no known impact on the overall test sensitivity, but only the decreased sensitivity for the S gene. Okay, so as I mentioned in my first session just now, you will likely observe the S gene dropout due to the 6970 deletion. The S gene dropout may signal the presence of the B11529 Omicron variant. So, for example, if you have a 3-target assay with operating frame 1AB, S gene, and also the N gene, you will have the operating frame 1AB and the N gene detected, but not the S gene. So you will have 2 results instead of 3.
Biến thể Omicron thì đối với biến thể Omicron, nếu như chúng ta có 3 target là S gene, N gene và ORF1 thì đối với những cái biến thể này thì chúng ta sẽ chỉ chỉ nhìn thấy tín hiệu ở S-gene, ở engine và ORF1, nhưng chúng ta sẽ không nhìn thấy tín hiệu ở S-gene. Do là các bạn có thể thấy là do là cái đột biến số 6970 deletion đấy thì nó ở vị trí S-gene. Thực ra là khi mà Thermo biến thể Omicron mà có đột biến ở đây ấy, thì cái S-gene này sẽ không phát hiện được. Ngoài ra thì nó sẽ không ảnh hưởng đến cái độ nhạy của xét nghiệm này. Xin lỗi, có anh chị nào hay là có các bạn nào có muốn tương tác trực tiếp không ạ? Anh chị có thể đặt thẳng câu hỏi với Dr. Christopher được không? Thì buổi thảo luận của chúng ta nó sẽ vui vẻ, sẽ thoải mái hơn ạ. Có anh chị nào muốn tương tác trực tiếp không ạ? There's one question from Phan Thị Liễu. Bạn ấy có hỏi là bạn có thể liệt kê giúp các thành phần của bộ kit taqpath CIVD kit được không? Okay, this question is, could you please tell us what is the components of the taqpath kit with CIVD mark here?
Okay, so this PCR kit is a very straightforward and easy to use kit. In the kit, you will have your assay multiplex. Okay, so this assay multiplex will have your primers and probes. You have your master mix, okay, and then you have your MS2 as your internal control. And of course, we also have your positive control in the kit. So these are the four main components. So for the PCR, you only need to add the assay multiplex, master mix, and water. So these are the three components for the PCR. Very straightforward.
Okay, thành phần của bộ kit này tôi nghĩ là cũng tương tự như thành phần của các cái bộ kit real-time PCR khác, real-time RT-PCR khác. Trong đấy thì có những thành phần chính là master mix, assay thì trong đấy thông thường là sẽ là primer probe, và thành phần thứ ba thì bao gồm control, có positive control và internal control. Đấy là những cái thành phần chính của bộ kit này, và tôi nghĩ là những thành phần này nó cũng tương tự như những cái thành phần chính của các bộ kit khác. Tuy nhiên là mỗi một bộ kit thì nó có những cái đặc hiệu khác nhau, và chủ yếu là nằm ở trong cái phần assay, tức là primer probe là chủ yếu, và master mix sau đấy. Can I ask you please, Chris, that's my question.
Yes.
Can you hear me? Yes. So you said that this taqpath kit have CIVD certificate, right?
Yes.
So since then you have this certificate?
Sorry, since when we do have this certificate? You
Yes.
mean? Okay, we actually obtained this certificate last September 2021.
I see.
Yes. For Vietnam. But we have launched this CIVD kit back in 2020, middle of 2020. So only in September 2021, we have the PCR kit being officially registered with Vietnam government.
Okay, so in September last year,
yes,
you have registered for Vietnamese government, right?
Yes, you are right.
I see. Câu hỏi của tôi thì tôi có hỏi là từ bao giờ thì kit này được cấp chứng chỉ CIVD, đấy là cái chứng chỉ có thể sử dụng trong chẩn đoán ấy. Thì tiến sĩ Christopher có trả lời là họ có được cái chứng chỉ này từ tháng 9 năm 2021 là năm ngoái. được cấp ở chính phủ Việt Nam. Another question. Bạn Lê Thiên Thanh có hỏi là S-gene dropout có ảnh hưởng đến độ nhạy của xét nghiệm RT-PCR hay không? Another question for you, Chris.
Yes.
So, the S-gene dropout, is it affect to the sensitive of the real-time RT-PCR or not?
Okay, so again, as mentioned, in general, the S-gene dropout will not affect the PCR sensitivity. Okay, when you choose a non-S-gene PCR test, for example, if you use the PCR test that target the E gene or the N gene only, then you will not have any sensitivity issue for that PCR kit. For example, for the taqpath kit, again, we have 3 targets. We have the S gene, the N gene, as well as the open reading frame 1AB gene. So with 3 targets here, okay, if you deal with the Omicron variant, S gene will not be detected. So only the 2 genes will be detected. So this result will actually give you an idea that, okay, this sample could be the Omicron variant.
Câu trả lời là việc không phát hiện được cái tín hiệu của S-gene thì không ảnh hưởng đến độ nhạy của phản ứng real-time PCR. Tuy nhiên là cái việc S-gene và dropout này thì sẽ ảnh hưởng đến việc đánh giá cái kết quả của cái phản ứng PCR này. Bởi vì là chỉ có 2 target là chúng ta có thể phát hiện được ở đây, đấy là N gene và ORF gene thôi, ORF1 gene thôi. Sau đấy thì chúng ta có thể nghĩ đến cái việc là giải trình tự theo như tôi hiểu là để tìm xem là có phải là biến thể Omicron hay không. Nhưng câu trả lời đây là S-gene dropout thì không ảnh hưởng đến độ nhạy của phản ứng PCR. Ngoài cái box chat này ra thì có anh chị nào muốn hỏi câu hỏi trực tiếp không ạ? Okay, another question for you, Chris.
Yes.
Có một câu hỏi từ xét nghiệm 29 hỏi là kit taqpath của Thermo có cách đọc dương tính là 2/3 tác nhân. Vậy nếu kết quả của real-time PCR chỉ lên 1 tác nhân, trong đấy không có gen S thì kết luận như thế nào? Âm tính hay là nghi ngờ nhiễm Omicron alpha? Okay, very interesting question, Chris, for you. For taqpath kit from Thermo, when there are 2 from 3 target gene is positive, then it will be positive conclusion. But in case there's only 1 target in 3 targets detected, then what can we say? How can we conclude? Is it will be negative conclusion or will be with Omicron or alpha suspected?
Okay, okay, thanks for the question. So for this one, okay, again, the taqpath kit, we have 3 targets, right? We have the S, N, and open reading frame 1AB. Okay, so let's say in the certain instances, you have only the N gene being detected. Okay, so the result will be inconclusive based on our interpretation, will be inconclusive. Okay, if 1 out of the 3 detected, it will be inconclusive. So what I would recommend you to do here, you should recollect the swab, okay, and repeat the PCR again. If you get the same PCR result with 1 out of the 3 target, then you can actually sequence the sample to find out what is the mutation that is happening in that sample.
Sorry, Chris, the signal was not very good. So could you say it again?
Okay, sure. Okay, so if the PCR result will be inconclusive. If you have 1 out of the 3, okay, it will be inconclusive. Okay, so let's say the S, N, and open reading frame 1 ABG, there are three targets, right? So if 2 of them are not detected, only 1, okay, you will need to repeat the PCR testing or recollect the swab sample. Okay, so after repeating the PCR, let's say if you still obtain the same result, 1 out of the 3, you can actually do a sequencing to confirm.
Okay, thank you. Theo câu trả lời của Dr. Christopher thì nếu như chỉ phát hiện được một các ghép trong 3 thì sẽ không kết luận được là âm tính hay là dương tính. Trong trường hợp này thì chúng ta có thể thứ nhất là lấy mẫu lại và lặp lại cái xét nghiệm realtime PCR này, hoặc là cách thứ hai là chúng ta sẽ phải giải trình tự cái mẫu này để có thể kết luận được. Thank you. Có khá là nhiều cái tin nhắn ở đây là có xin ban tổ chức tài liệu sau hội thảo ấy, thì có đại diện nào bên Thermo các bạn SISC các bạn có thể trả lời được không ạ?
Dạ vâng, thưa quý vị, thì nhân viên SISC chúng tôi sẽ liên hệ trực tiếp để chuyển cho quý vị các tài liệu này sau khi nhận được email từ các địa chỉ email đăng ký ạ.
There's another question for you, Chris. Câu hỏi từ bạn Dương Ngọc Thúy. Nhờ chuyên gia chia sẻ về giải pháp giải trình tự sang G phát hiện biến thể Omicron. Could you please share with us more detail about Sanger sequencing for Omicron variant detection, please?
Yeah, sure. Okay, if you would allow, I would like to share my screen for this one. Okay, just give me a moment. Okay, I hope you can see my screen. Yeah, can you see my screen now?
Yes.
Okay, so this is the Sanger sequencing solution for SARS-CoV-2 research. Okay, I think we will share the link with you shortly after this webinar. So under this webpage, right, you can actually find a lot of tools here. For example, you can visualize the SARS-CoV-2 genome and other primers. Okay, so let me zoom out. I hope you can see. So you can see visualize SARS-CoV-2 genome and other primer. So once I click this, I will come to this page. Okay, so this page, we will have all these available tracks here. Okay, you can see this is the SARS-CoV-2 genome here. Okay, then when I click the B11529, this is the Omicron variant. So this will show you where the location of all the mutation here. So you can see. So these are the amino acid change here. All right. So you can actually see where are they located. So it depends on whether. So just now we have 4 protocols, right? We have, of course, the S gene protocol. So you can sequence the whole S gene of SARS-CoV-2. Then you can find the location of this here. It is very comprehensive. If you want to look for the primer, you can also come back to this page. You see search SARS-CoV-2 genome and other primers here, this one. If you click this, you will come to this page. Okay. So here, then you see I could select the alpha or other strains. Okay, so this is the delta one, B1617.2. So now I'm selecting the Omicron. Okay, so the gene here, I would select the S gene. Okay, then here the S gene, I will actually key in the known amino acid change. All right. Then this is based on these genomic coordinates. Then I will be able to have the primer being generated for you. Then you can place order from us. Then it will save you time to actually to sequence only the certain portion from your S gene. Is it quite clear for you?
Yeah.
Yes.
I see. Yeah, I see here still there are a lot of questions.
Yeah. Okay, let me stop sharing my screen for a while. Okay, let's say if you cannot answer your question today, I'll be more than happy to send my answer to you through emails.
So yes, you can lift your screen. There's a question from Chung Sun Lei. So is there a product that can differentiate between SARS-CoV strains by qPCR?
Okay, so for this question, I will also want to share my screen here also. Just give me a moment.
I think another similar question is in Abdington Phước. He asked if we want to confirm the Omicron variant by real-time PCR. Is it possible?
Yes, it is possible. Okay, so if you look at this website from Thermo Fisher, we have our mutation panel. Okay, so this mutation panel covers all sorts of the variant here, as you can see. Okay, then when I scroll it down, okay, we actually have these are the workflow. Okay, then you will have let me scroll down further. So just give me a moment. Okay, yep. So this is the new Omicron variant mutation assay that we have here. Okay, so we have the G339D, Q493R, T547K, this one, and also the T13195C. Okay, so how do we actually get this mutation assay is from this paper actually. Okay, you can see this paper. This paper, they actually created 3 Omicron specific marker and also 1 Delta marker to help you to differentiate between Omicron and Delta. Okay, so under this page, you can also find the mutation here. So for example, let me just find an example for the Omicron variant. Yep, for example, this one. So this is from Open Reading Frame 1AB gene. This is the mutation, and this is the for Omicron, okay? And this is the assay ID. So you can order these. These are the mutation for Omicron. We have another one for Omicron also, for example. Yep, so here. These will be the assay that helps you to detect the Omicron variant, this one and this one. So again, this paper, okay, they set up these 4 target qPCR tests just to help differentiate and confirm the Omicron variant as well as the Delta variant. Okay. I hope I did answer your questions. Is it quite clear?
Vâng, các anh chị chắc là các anh chị đặt câu hỏi về cái này thì chắc cũng chuyên sâu về sinh học phân tử rồi ạ. Thì Chris vừa mới giới thiệu với chúng ta cái trang web của Thermo đây, và trên trang web này thì chúng ta có thể xem được rất là chi tiết các cái vị trí đột biến của cái biến chủng, của từng biến chủng một có những cái đột biến gì, và thậm chí là họ đã có sẵn những cái trình tự để ở trên này. Và nếu mà để xác định được cái biến chủng Omicron này thì không phải là sử dụng bộ kit taqpath mà họ đang giới thiệu ở đây, mà là những cái bộ kit đặc hiệu khác của Thermo. Họ đã có thiết kế để xác định được biến chủng Omicron. Vâng, còn cái giải pháp taqpath realtime PCR như tôi hiểu thì chỉ là dựa vào cái sự dropout của gen S để dự báo là đấy có thể là sự xuất hiện của biến chủng Omicron thôi, chứ không phải để xác định đây là biến chủng Omicron hay không. Okay, another question from Văn Ngãi, from Ngãi Medlatech.
Yes.
Bạn Ngãi có hỏi là tôi muốn hỏi điều kiện được sử dụng xét nghiệm giải trình tự gen là gì? Nếu chúng tôi sử dụng hệ thống giải trình tự của Thermo để định danh biến thể Omicron thì có được công bố hay không? Muốn được tự công bố thì cần điều kiện gì? Ngãi ơi, chị nghĩ là cái câu hỏi này chắc là cũng không phải phụ thuộc vào Thermo nữa. mà nó sẽ phụ thuộc vào quy định chung. With this question in general, when should we use the sequencing technique for this?
Okay, when should we use the sequencing technique, you mean? Okay, so in terms of normal clinical testing, all right, our taqpath kit would already help you to somehow identify the presence of the Omicron variant. Okay, so it depends on your downstream work. When are you going to use it for publication or research? It depends on how you want to manage these patients infected with Omicron. Not necessarily you need to sequence all of them, okay? But if the patient with the Omicron variant demonstrated some really interesting symptoms, okay, or any clinical complication, then you can actually sequence the sample to find out more. And it is also related to the cost also, how the cost, if you want to save some cost, okay, of course the qPCR would be enough. If you have additional budget, then you can actually proceed to do the sequencing or for research purpose or for publication purpose.
Ngãi ơi, em có ở đây không?
Dạ em có nè.
Ngãi em ơi, em có thể show lên để tương tác trực tiếp với anh Christopher. Em câu trả lời vừa rồi của anh ấy em có ý kiến gì không?
Em không nghe rõ.
Tức là theo chị dịch qua nhé, thì theo như anh ấy nói thì nếu mà để sử dụng cái hệ thống giải trình tự gen này thì cũng tùy vào mục đích của mình là gì, và nó cũng tùy vào cái chi phí của mình. Mình muốn chi phí cho nó như thế nào, bởi vì chắc chắn là giải trình tự thì nó sẽ đắt hơn là realtime PCR. Tất nhiên là giải trình tự thì nó sẽ mang lại nhiều thông tin hơn cho mình. Thì chi phí của mình là gì và mục đích của mình là gì nữa, mình dùng để nghiên cứu hay là mình dùng để chẩn đoán, thì mình có thể quyết định sử dụng cái phương pháp nào. Và cái câu hỏi của em là nếu sử dụng hệ thống giải trình tự của Thermo để định danh biến thể Omicron thì có được công bố hay không, đúng không? Tức là có được public thành bài báo hay không nếu sử dụng hệ thống của họ?
Em làm rõ nhiều hơn cái ý của em ấy. Thực ra là bây giờ trong cái ngành y của mình ấy, mỗi cái xét nghiệm của chúng em hay một dịch vụ nói chung ấy thì đều phải đăng ký với lại Bộ Y tế, Bộ Y tế phê duyệt. Vậy thì cái xét nghiệm này có cần phải phê duyệt của Bộ Y tế hay không? Ngoài ra thì việc là mình các cái xét nghiệm Covid thì đều phải là qua thẩm định hay khẳng định của Bộ Y tế hay là Viện Vệ sinh Dịch tễ. Vậy thì cái xét nghiệm định danh này có cần phải thẩm định lại hay không? Thì lúc đấy mình mới có khả năng là mình làm xét nghiệm ra thì có được công bố đến phòng xét nghiệm chúng tôi phát hiện được cái chủng Omicron này hay không, chứ không phải là phòng xét nghiệm hay là phòng xét nghiệm nào cũng cứ phát hiện ra là công bố được cái giá trị và độ tin cậy của nó nữa. Ý em là như vậy.
Chắc là cái này SISC phải trả lời. SISC các bạn có ở đây không? Lan Anh ơi.
Dạ vâng, xin chào anh Ngãi. Về cái câu trả lời này ạ, thì chuyên viên của chúng em sẽ liên hệ trực tiếp với anh để tùy thuộc vào là anh sử dụng giải pháp realtime hay là Sanger hay là NGS, thì các anh chị bên bán hàng sẽ hỗ trợ trực tiếp với bên mình không ạ? Bởi vì thời lượng của buổi hội thảo hôm nay chúng ta cũng đã quá giờ khoảng độ 7 phút rồi ạ. Thì
Dạ vâng ạ.
và sẽ liên hệ lại trực tiếp với anh về các thông tin liên quan ạ.
Dạ ok ạ.
Chắc là chị cũng thấy một số câu hỏi cũng rất là nhiều câu hỏi đấy Lan Anh ạ. So
hôm nay thì cũng hơi
do we still have time or we have to stop here for tomorrow?
We have a lot of questions coming in, but the session for today, the time is almost up. Christopher, what do you think? Would you like to answer more questions, or we should wrap it up and continue tomorrow?
Okay, I think we can collect those questions so that I can reply them personally, separately through email. What do you think?
Yes, that would be very nice. We will collect the information.
Yes.
Dạ vâng.
Thank you.
Vì thời lượng cho buổi hội thảo hôm nay đã kết thúc thì chúng tôi xin chân thành cảm ơn tiến sĩ Phạm Thu Hằng và ông Christopher với buổi thảo luận sôi nổi vừa rồi. Các cái câu hỏi của quý vị mà chưa được trả lời thì chúng tôi cũng sẽ chuyển đến ông Christopher để có văn bản trả lời lại cho quý vị. Công ty SISC và hãng Thermo xin chân thành cảm ơn sự quan tâm của quý vị đã dành cho buổi hội thảo ngày hôm nay. Tôi hy vọng thông tin và giải pháp của hãng Thermo chúng ta đã góp phần nào đó vào công cuộc phòng chống đại COVID-19. Chân thành cảm ơn hãng Thermo và tiến sĩ Phạm Thu Hằng đã hỗ trợ chúng tôi tổ chức buổi hội thảo ngày hôm nay. Vào cùng giờ này vào ngày mai thì chúng tôi cũng có một phiên thứ hai của buổi hội thảo này, nhắc lại các nội dung trên và tiếp tục mục thảo luận. Chúng tôi xin gửi lời cảm ơn chân thành tới các y bác sĩ, các kỹ thuật viên và các chiến sĩ trong công cuộc chống dịch đã vất vả hỗ trợ người dân trong thời gian vừa qua. Chúng tôi xin kính chúc sức khỏe và chúc quý vị đại biểu một năm mới 2022 đạt nhiều thành tựu mới để chúng ta cùng nhau chung tay sớm đẩy lùi dịch bệnh COVID-19. SISC xin chân thành cảm ơn quý vị đã quan tâm đến buổi hội thảo ngày hôm nay. Chúng tôi xin kính chào quý vị và xin phép được kết thúc buổi ngày hôm nay ở đây.